All right, next thing we got to talk about here, guys, proper lab report format. All right, so that is, what do we have to put on a lab report? How does that need to go? What order does it need to go in? Okay? And everybody in our science department here at the high school has the same idea of a lab report. We want to see the same things, and we want to see them in the same order, right? So when you learn this in Science 10, you can apply this in biology, chemistry, physics. Doesn't matter which science you take, we're going to want to see the same stuff done in essentially the same way, right? And that's because this is the scientific method, right? I mean, there's slight variations on some things when you get to university, but for the most part, this is the way it's going to go. You can't have a lab if you don't have a problem that you're investigating. So that's the first thing that'll appear on a lab report. When you get your lab report, or you get your lab given to you, there'll be a problem that we're investigating. Okay? I'll have it written out in my words. What you will have to do is rephrase it and put it in your own words. And it needs to be a question. It's like Jeopardy. Okay? Right? You have to phrase your answer in the form of a question. This is how you do a problem. Right? So the example that we're going to work through here is, what is the effect of light on plant growth? It's incredibly simplistic, and thus it's a good example. Okay, so the problem we're investigating in this hypothetical lab is, what is the effect of light on plant growth? Okay, once we know what the problem is, then we've got to come up with, what are our variables? Okay, there are three kinds of variables. Manipulated variable, and there can only be one of those. Okay? In any experiment, you can only have one manipulated variable. If you have more than one, you'll never know which of them caused the change that happened in the lab. Okay? Because there's no way to separate the two. Right? So you only ever have one manipulated variable. You'll have responding variables. There may be more than one responding variable. Okay? When you make a change to something, it's possible that that could have several effects, and you would be watching for maybe a couple of things. Most of the labs we do, it's going to be one responding variable. Okay? The last kind of variable are the controlled variables. And oddly enough, I think those are the most important ones, because they're the ones that can make a lab invalid if they're not controlled. That's why they're called controlled variables. They are the things within the environment that could affect your experiment if you don't keep them constant. Okay? So, manipulated variable. Okay, it's the part of the experiment, okay, changed on purpose by the experimenter. Design. Sorry about my penmanship. I thought I had typed this up, and I didn't, and then I started typing it this morning, and I didn't get it all finished. So yeah, my grade 6 teacher told me, your penmanship's awful, and I told her, everyone's doing stuff on computers now. No, that was back when computers were gigantic things, but I ended up being right. I kind of like that. Okay, so manipulated variable, the part of the experiment changed on purpose by the experimenter. In our hypothetical light on plants experiment, the manipulated variable would be the amount of light given to the plants. So you can often look at the problem, and the problem will tell you what the manipulated and responding variables are. Okay? Our problem said, what's the effect of light on plant growth? So light is the thing we're changing because we want to see its effect on the responding variable, plant growth. Okay? So you can almost always look at the problem and know the first two variables. Okay? So a responding variable then is the thing that's going to change as a result of what we did. Okay, so it's whatever changes as a result of the changes made by the experimenter, or as a result of the manipulated variable. Okay, in this case, it's the growth of the plants. Now, in a good lab report, you would probably want to specify how you're measuring the growth of the plant. Okay? And in fact, you may measure it in more than one way. Okay? You could have a very simple method by, recording the height of the plant if it's a plant that grows upwards. But you might also want to record the increase in its mass over time, counting the number of leaves. Okay, maybe if it's a plant that produces fruit, okay, recording the amount of fruit that it produces, okay, and things of that nature. There's many different ways to record your responding variable, and it's often a good idea to tell people when you're identifying it, here's why it's the responding variable, and here's the things that are going to be its indicators. Okay? So when you're writing up a lab report, you identify the variable, and then you tell me why. Okay? So light is the manipulated variable because it's going to be changed by the experimenter in this way. Okay? The responding variable is the growth of the plants because we think plants depend on light, and here's how we're going to measure that. Okay? And then lastly, okay, like I said, I think the most important variables are the controlled variables. Okay? They are other factors, and they're usually environmental factors, okay, that could affect the outcome of the experiment if they're left unchecked. So if I'm doing an experiment on plant growth, I probably have some understanding of what things can affect the growth of a plant. I'm trying on purpose to test light. So what things should I keep the same for all of the plants? Candy? They should all get the same amount of water. They should all be in the same container. They should all be the same kind of plant. Soil or a fertilizer if we choose to add that. Okay? Anything else that can affect the growth of a plant needs to be the same for all of my test subjects. It does me no good whatsoever to record, to test this experiment on a spruce tree, a tomato plant, and a carrot. Okay? They all grow at different rates. I'm going to see virtually no change in the spruce tree because they grow very slowly. Okay? And I'm going to see the carrot grow rapidly. Okay? But it grows mostly downwards. Okay? And, I mean, it's got, you know, the downward growth of the part you eat, the tuber. Okay? And then it's got the foliage on top, which generally doesn't get very tall. Okay? It doesn't do me any good to compare those because they grow differently. Right? I would want to do this test on all of the same kind of plant, all with the same amount of soil, okay? Same temperature too. Right? I don't want to have them all in different places and have one growing at like 2 degrees Celsius and another one growing at 20 and another one growing at 50. Okay? They're definitely going to have different rates of growth because the temperature is different. So controlled variables have to be exactly that, controlled. Okay? They could affect the outcome of the experiment if we don't. Generally, those become our sources of error. Okay? If we find something was weird in the lab, it's usually because we missed a controlled variable. And then you would say in your sources of error, it was realized later that the following variable, you know, something happened with it. A light bulb burnt out on plant number 2. Okay, so the results from plant number 2 are invalid. Okay, because it did not receive the proper amount of sunlight. That's beyond your control. Okay, happened within the lab. What do you do? Okay. All right, so like I said, the examples here would be same plant, same soil, same amount of water, same temperature. Okay? Anything that could affect the experiment. Okay, when we do our lab across the hall here, obviously all the samples will be the same temperature. Okay, because temperature affects things like solubility. Okay, and then solubility could affect things like conductivity and pH. So yeah, temperature is going to be an important control variable for that one. Okay, we're also using the same amount of everything. That's why we're using a depression plate. Okay, we can only put so much of everything in these little pits. Right? All right, after we've identified the variables, we have to come up with our hypothesis. The hypothesis and the conclusion are probably the two most important parts of a lab write-up. Okay, your hypothesis tells people what you think the cause of the problem is, or the answer to the problem is, or the relationship is between the two variables you're studying. Okay, it tells them that. It briefly describes your experiment, and it tells them your predicted results if your idea is right. Okay, So a hypothesis always contains three words: if, and, then. If this is true and this is done, then this will happen. Okay? Always goes like that. If, and then. Okay? It's a statement that communicates, okay, the answer or relationship in the problem, describes briefly the experiment, and outlines the expected findings. Okay, and you can see there that I've kind of, this is the if part, this is the and part, this is the then part. Okay? Okay, so the if part describes describes the relationship, or it answers the problem. Okay, something along those lines. So if this is going on, or if this is the cause of this, something along those lines. And the key word here is brief description of the experiment. Don't go into detail about how exactly you're going to perform that. That's what the procedure is for. Okay, this just briefly outlines it. Okay, the then part outlines the expected results. This is what I'll see if I'm right. We don't say it like that because we try not to use I, we, us in a hypothesis. Okay. Okay, so the most common mistake I see in a hypothesis in a lab report is someone just telling me what's going to happen. Okay, they go like this: I think the plant that gets the most light will grow the most. Well, that's really only one part of the hypothesis. Okay, the then part. You've predicted your findings, but that's not what the hypothesis is all about. It's part of the hypothesis. Okay, you have to be putting yourself out there, essentially. You have to give yourself the chance to be wrong. Okay, that's what science is all about, right? In a real scientific endeavor, most of your hypotheses will be wrong. That's how science progresses, by finding out what isn't right. Okay, Thomas Edison was one of these guys when he developed the light bulb. Okay, he tried like 300 different models to develop the light bulb, and someone asked him, well, you kept trying and trying and trying, did you ever consider yourself a failure? And he said, no, I came up with 299 ways how not to build a light bulb. Okay, and you honestly, with most hypotheses in the real scientific community, you're going to be wrong, but that's how you learn, and that's how you progress towards what's correct. And I don't mean by wrong completely wrong. Okay, you're almost never completely wrong, but you almost always find something that isn't quite true. So you might conditionally accept a hypothesis. Okay, and say, this part certainly seemed to hold true, but there was this issue, okay, here, so further experimentation is required with the following modifications. That's what your conclusion is for, is to say, I accept this hypothesis for these reasons. The following things showed up, and those will be further studied. Okay. So for our plant lab, right, the if part's going to say what we think the effect of light is. Right? That way we can be right or wrong. The and part is going to talk about what we're going to do, and the then part will tell us the expected findings. So it'll sound something like this. Hey, if plants require light for growth, so I've said they can't grow without it. That's the if part. Plants require it for growth. And 5 identical plants are given different amounts of light. That's a brief description of my experiment. I'm going to do 5 plants. They're all going to get different amounts of light. I'm not saying how much light, just different amounts. Okay? Then the plants that receive the most light will show the most growth. Okay? It is now entirely possible for me to be wrong in this experiment because I have said, here's what I think the relationship is, and here's what I think is going to happen. Okay? So the hypothesis is usually the thing that requires the most sort of practice and most thought, okay, when you're writing up a lab report. And honestly, you know, when I read a hypothesis, if the hypothesis is good, generally the rest of the report is good too, okay, because a person has really thought about what they're doing in the lab. Okay, when a hypothesis is really short or in the wrong format or something like that, generally the rest of the lab follows that. Okay, and then how do you have a good conclusion if your hypothesis was crappy to begin with? Right? It's really difficult to say, well, you know, my hypothesis is acceptable because, well, I didn't say anything in it, and therefore I couldn't be wrong. Okay? Well, then what was the point of doing the experiment? Okay? All right, this part here we kind of have. If you drew the stuff I have in green here, this is just kind of saying that over again. Okay, just saying in the if part. Okay, that's what's the answer or what's the relationship or the cause. Okay, the and part briefly describes the experiment. Okay, and the then part is what you expect to find if you're right. Okay, okay. From there, you move to materials. What do you need to perform this lab? For you guys, those are always going to be written out for you. Okay, because when I give you your lab report template, it's going to say materials, and then there's going to be the list of materials. Okay, it's always just going to be there, so you guys really never need to worry about it. Okay, even for the lab that you're going to be designing, we've already talked about what the materials are. The unknown compounds, a depression plate, a conductivity test, or pH paper. We know what the materials are. It's not something that you guys, at least in Science 10, will have to think up. All right? But when you get to the higher levels, okay, like university, and you're completely designing and performing an experiment, you do have to come up with what materials are going to be best for this. Okay? In an experiment like this, deciding what kind of plant is most appropriate is a pretty important step. Okay? It just doesn't really show up anywhere in your data here, but it's still an important step. If I want to test the effect of light on a plant growth, I got to pick something that grows quickly, not something that grows slowly, unless I want to be doing this experiment for the rest of my life. Okay? I probably just want to do this experiment over a few weeks. Okay? So something like beans would be something that's good. All right? Because within 90 to 100 days, they've gone through their full life cycle from germination to the production of fruit, and they're done. Okay? Everyone follow me there? Beans are fruit. Okay? Beans are fruit. Anything that comes from a flower is fruit. Remember that, because we think lots of things are vegetables that are fruit. Tomatoes. Fruit or vegetable? Fruit. Cucumbers. Fruit. Peas. Fruit. Come from a flower. Okay? Then it comes from a flower. It's fruit. Flower, fruit. Okay? Flower, fruit. All right. That was off topic. Anyway, okay, materials. Sometimes you won't even write C sheet anymore because I'll have them on the template. That's the beauty of Google Classroom. They're going to already be there. Okay, every time we do a lab, you're going to get a file that says, you know, properties of compounds lab with your name, and the template will be there. Basically, you fill it in. Okay, you write in it'll say problem, and it'll be blank. You'll write in the problem. Hypothesis, blank. You write it. Okay. Variables, manipulated, responding, controlled. They'll be blank. You just fill them in. Okay. All right, so materials, they'll already be on there. Then you got your procedure. Normally, that will also be written for you, okay? There's really only this lab, maybe one other if we have time for it, where you actually have to come up with the steps. Most of the time, it's going to be cookbook. Here are the steps, okay? Do this, then do this, then do this, all right? It's a detailed set of instructions, okay? They have to be explicit. And by that, I don't mean they contain bad words. Okay? I mean, they are extraordinarily clear. That is actually the strict definition of explicit. Okay? They are extraordinarily clear. Assume the person following your instructions is a complete moron. Okay? If you make any assumptions, they'll do it wrong. Okay? You have to assume that. And don't be insulted by that, because you'll be following my instructions all the way through on most of your labs. Okay? But you have to assume, okay, that the person following them has no idea what they're doing. Okay? So be very, very clear. Go and get the required materials from the front table. See materials list. Okay? Perform observable properties test. Look for the following things. Color. Okay? Make note of this in observations chart. Okay? Etc. Be explicit. Be very, very clear. How do I do this? All right? So they are for preparing and performing, okay, the experiment. Okay? Then you're going to have observations, your data. Okay? And the data is going to contain graphs, charts, pictures. Okay, if it's that kind of a lab, when you're doing a plant growth lab, you may very well take pictures of the plant every day or every week or whatever it happens to be. Okay, as well as making quantitative observations. That would be your measurements that are actually numbers. Okay, pictures would be a qualitative observation. Which one? Anecdotal means like you wrote a story, not really a fictional story, but it was observed today that the plant was, a little darker green. Or, the leaves were a little droopy today, something like that. Okay, anecdotal notes. Okay, we'll talk about analysis, conclusion, and sources of error tomorrow before we get started. It won't take us long to get through those. Okay, I will post tomorrow's quiz after school today or sometime this afternoon.